Welcome to Under the Microscope podcast, where we spotlight materials and nanoscience. Under the Microscope is the flagship podcast of the Science Talk, and here we explore the fascinating world of materials and nanoscience. Our goal is to provide a stage where scientists can communicate their work and interact with the public. With that in mind, with every episode, we introduced you to a scientist working in the field of materials and nanoscience. Join us as we delve into the groundbreaking research, innovative discoveries, and the inspiring stories of scientists making waves in this field. Welcome to Under the Microscope podcast of the Science Talk. My name is Pranoti Kshirsagar. I'm your host, and today we have with us Humeyra Caglayan, who is a professor at not one, but two universities in Europe, one in Netherlands and the other in Finland. In Netherlands, she is a professor at the Eindhoven University of Technology, and in Finland, Tampere University. Humeyra is going to tell us about this very exciting project called 3D Microscopy, funded by Jane and Aatos Erkko Foundation. The funding call supporting the 3D Microscopy project is Future Makers. This project started in 2023, so it's been running for 2 years now, and it will go for another 1 year. And of course, what better podcast to talk about 3D microscopy than Under the Microscope? Please join me in welcoming Humeyra. Hi, Humeyra.
Hello.
How are you doing today?
I'm doing great. Thank you. How are you?
I'm good. I'm so excited to talk about this project, 3D microscopy. I don't know if you remember, but the first time you told me about it, my eyes just went like, wait, what? And then what? So tell us, what is the scope of this project? In super simple words, please.
Yes, sure. I think everybody knows about the imaging and especially biomedical imaging. That's super important in our life. The need on the biomedical is that we have to be looking for 3D images, but we cannot sacrifice a lot of time to collect this 3D image. And our project is all about getting these 3D images in a snapshot so that we can get the best quality images, volumetric images for either research purposes or for treatment purposes.
What are you imaging here?
So we are imaging from cells or any kind of large biological samples as well, can be organs. That's of course quite tricky because what we really do in current technology is that we have to scan them slice by slice, and that's not ideal. So if we would like to have an image of a living cell, for example, we need to do it very quickly so that we can see the whole functioning in a very short amount of time. One good example is a zebrafish. Zebrafish is, of course, a fish that is quite often used in biomedical imaging, and we really want to image the zebrafish so that we can see how its brain functions or how its retina or heartbeat, that we could see those things. So what we are imaging is that volumetric image of zebrafish, but also in a very short amount of time so that we can record it as well during that time without harming the cells or biological samples.
So, okay, wait. This is completely a new concept also for me now, because for my PhD, I did transparent microelectrodes for electrophysiology, so simultaneous electrophysiology and optical imaging. And back then, when I wanted to see the interface or interaction between the cells and my samples, like my graphene sample, for instance, I had to freeze it. I had to do like the dye and all of that. And of course, at the end, the cell was frozen, so it was dead, like a cardiomyocyte or so. And we got these beautiful images where we could see the insides of a cardiomyocyte, which is a heart cell, and how it was interacting, or rather how it was sitting on my electrode. So what you are telling me is that with your technology, we won't have to kill the cells. The cell can just exist. It's living while we are still able to get a 3D image. If you would have told me that we will just get a 2D image, I would already be super happy. But what you're telling me is that we'll get a 3D image.
Yeah. Well, of course, it's a challenge, right? So because in such kind of a setting, as you mentioned, we either have to freeze the sample because we need a lot of time, and that's why we need to scan step by step each focus point and then get the image. So that's the limiting factor in terms of time. So you have to focus to a certain plane and then get the perfect image, and then you have to go another one and do it again and again and again along the body of the sample. In our technology, what we innovate is that we can extend the depth of focus along the region that is needed for the whole scan. So we can get it at a snapshot the image. So we do not need to scan at each focal point, but we can get the whole image at a perfect focus. So that is the innovative part.
How? Okay. This is like, okay. If you're talking about microscopy, the first question that comes is what is the resolution, right? So,
Yeah.
can you tell us about what resolution are we talking about and what area are we talking about with the 3D microscopy, like the 3D map of sorts? How big can you do? How quickly can you do? And what is the resolution? How clear will the image be?
Yeah, I think the challenge with the microscopes, right? So you can either sacrifice the resolution or how much depth you could go and so on. So they're all related concepts within each other. What we are doing is that we are innovating this novel optics that we call meta optics. And this with meta optics, we are extending from a couple of microns to 300, 500 microns. So then we can have this whole image in focus. So we are doing it, of course, within the availability as well, also the software of the reconstructions that we develop so that we can bring all together within the correct image group.
And you will do the imaging and the cell can just exist, right? It can just
Yeah.
go and do whatever.
So aim is that to scan the whole zebrafish at a snapshot. So giving that dimension so we do not have to do a lot of scanning. Of course, for the perfect imaging, I think if you prefer higher resolution, you might go into 1 or 2 times of scanning, but still we are talking about maybe milliseconds of whole scanning. So already quite good.
Wait, did you say milliseconds for the
Yes.
entire zebrafish? How big is the zebrafish?
Well, zebrafish can be about 500 micron by 500 micron. You can think of the range of the image that you would need when you're imaging a zebrafish.
Oh, wow. So for people who are not familiar with these dimensions, that is half a millimeter by half a millimeter being scanned in fraction of a second.
Yeah, indeed. And why we want to do it in a very short amount of time is because brain activities can be quite fast. Currently, I think the best one being recorded is at 1.5 seconds intervals. So we can only see the activity every 1.5 second, but you would like to see what happens in between these 1.5 seconds. So that's why we are calling this basically like a snapshot 3D.
Snapshot 3D imaging. That is brilliant. That is so cool. Because that was also going to be my next question, right? Because living things like to move. And I remember this from the cardiomyocytes, which are the heart cells, which form a carpet on a flat surface and they're beating like a heart. If it is not a snapshot, that just makes it more complex because then the image is blurry. So, what is the size of the data then? Because I'm just thinking if we want to record the neurons talking to each other or communicating with each other in the brain, because that was also one of the things why we wanted to have transparent microelectrodes. That is the closest I have as a comparison to understand your work and the application of your work in the living systems. The neurons, when they are firing and then when they are communicating with each other, neurons are the brain cells for people as a reminder, and they're firing very quickly. And you want to be able to understand and image it, visualize it, not just record the signal, but also be able to visualize it. And that is something that your project, the 3D microscopy and the snapshot 3D microscopy, is able to give. So, what kind of images are we looking at?
We are working on 3 or 4 different colors in the RGB range. So red, green, and blue. We are talking about different color images that we could collect. But of course, as we look at, especially for this kind of brain activity, then we would see some firing up of the neurons. Then it is like a little bit of more black and yellowish kind of colors. These are kind of the images that we could record. This gives us quite an opportunity to see in a short amount of time how these kind of activities happen that people then take this to another level for treatment and so on. But for us, the ideal is that we will get the most data possible within a short amount of time for future use of any biomedical purposes. And here we have to work both on the hardware and the software part. We have to bring them together. That's why you mentioned about the data. It's an important part as well. We cannot just give a raw data. So there we have to really create the image that can mean something for person that is looking at it. That's why we also then creating this image reconstruction part, software part, that one can really turn around this 3D image, look around which direction that you want to look and so on. Imagine that you have this kind of 3D volume image that you can turn around with your mouse. You can look at different directions. Could be quite useful, hopefully, to also to bring it any of the doctor's office in the future.
And that is the future that we see or want to see on TV shows like Grey's Anatomy, for instance, where Dr. Shepard doing the brain surgery and then they're like, oh my God, where is the tumor? I want to be able to visualize the tumor. Where is it? There comes Humeyra and her team with the 3D microscopy. Like, here you go, you have the 3D image, and now you're able to look at it and visualize it and move it around so that you only cut the tumor out and not the brain.
Yeah, indeed.
I'm not sure if that is the kind of dramatic application you had in mind, but well,
Yeah,
imagine it.
visualize it really nicely. Yeah, indeed. So if you could bring it to the surgery room, that is the ideal. They can make sure that they have this perfect samples in the surgery. That's the ideal case, of course, for us.
That would be
Yes.
perfect, right? It would be as less invasive as possible, which is great for the doctors, it's great for the surgeons, it's great for the patients as well. That is brilliant. So you also mentioned the software and the hardware part. I know you are an amazing scientist. You are a professor at two universities, two really big shot universities, but I'm assuming you're not doing this alone.
Oh yeah, that is a lot to do alone. So we are doing it actually as three different teams. So my team take care of the optical setup. We are mostly an optic photonic experts. So we are developing optic setup of the microscope and the novel optic part that we are developing mainly so that we could achieve this depth of focus that is enhanced. We have Johnson and Anil with me, my postdoc and my PhD student. And then for the software part, we are working with Atanas Kochevs and Erdem Shahin from another faculty. And they are the one who are developing the software part. They are also together with another postdoc, Yanni, and they are imaging experts. I call them sometimes that you're doing magic with images. Another partner is in the biology department, biomedical department is Temo. And Temo is the person who brings problems to us. So from biomedical imaging, he is the one who brings the problems and we solve it. So yeah, we are quite a big team that interacts from different parts. So we are both looking from perspective of optics, photonics, as well imaging parts.
That sounds wonderful. And I'm a big fan of major disciplines, different teams of different experts working together, because I think that is the key to innovation. The project has been running for 2 years and it will go for another 1 year. So can you share some initial results with us? How close are we to have this technology on this TV show, Grey's Anatomy, or in real life?
Yes, it's been 2 years and we have been quite busy trying to make this microscope setup. And now we have this setup and trying to make this working in the lab. So we have developed this novel meta optics parts. Initially, we showed how in theory it will work. This meta optics parts together with the image reconstruction. We had the hardware completed. So now we are making tests together with the software. We are using simpler biological samples to test our setup. This has been the work for us for 2 years. It's been quite a lot of effort from all parties. In the third year, now we're gonna really want to test this in a zebrafish, for example. That's the ideal. And once we can show all this, what we really want to do is more on the packaging, because now overwhelming to bring it to a doctor's office or surgery room. So what we really want to do is that to make it more user friendly so that they can do it without us in the room so that we don't have to go each room. And this can be quite be used by hopefully many places. Ideally, of course, we do not just want to leave it in any kind of biological samples, but as you mentioned, it can be used for tumors as well, so that it can bring it to the surgery room, so that we can take it to any places in the world. So be useful for the whole society at the end.
That's good to know. I can talk to you for hours and hours. I have so many questions for you, but I'll ask you one last question and then I will let you go because this has been fantastic. So, what are you most excited about in the next 6 months of this project?
So far, I have seen so much nice images from our setup. What I'm really looking forward to see is a real live sample image so that we can really record how it changes within a certain time. The images taken from our microscope. That's the final things that we are looking in this project. Now I'm also looking forward that we had an extension project of this project, exactly as you mentioned, to the surgery rooms with partners in the United States. So I'm also excited to start that project. That is to work with even also pathologists on this and what are their challenges and how can we solve or modify it using our microscope. So are the next big steps for us in the next 6 months, 1 year.
So next season of Grey's Anatomy will have your technology.
I hope so. Maybe it's been quite challenging for us to really think around it at each point of how to really improve it, because there was quite a struggle to have to balance the resolution aspect of it and like depth of field aspect, and then also make it compatible with the biological samples. This was all each point a different challenge for us to think. And one simple example that I always give is that it was quite challenging to think even to make a image 3D. Of course, you can always get Z images, but to make it at a snapshot, we really mimic there the human perspective. For example, people understand image if it's a 2D or 3D, and we mimic that right now. What they really do is that, are you familiar how people understand if it's a 2D or 3D? So what we do is a very basic human instinct. We turn our head. So we try to understand if it's a 3D or not. And that's actually what we're also mimicking with our this so-called novel optics. We are mimicking so that we are collecting it from different angles so that we could collect 3D. So it was quite challenging the way from looking from different optics, different software, and then trying to mimic also some of these things from nature and then implement it to really a biological sample. So now I'm really excited to see in the last year how we bring it to this whole thing together. But
Absolutely, yeah. And you also mentioned the meta optics, and I would love to get into more details of what do you mean from the physics perspective? What do you mean by the meta optics? How exactly are you doing it? That would be really cool for me, definitely to understand, and for everyone else also to understand. Of course, the application and when all of this comes together, this is great, but also the physics behind how exactly you did it. What you also mentioned with looking from different, like we tilt our heads, we move around, and that's exactly what your optics is sort of doing. So yeah, that would be awesome. Perfect. Thank you very much, Humeyra. Excited to talk to you again with the images and the 3D maps of sorts that you will have as the initial results or the next results. And looking forward to speaking with you again. This has been wonderful. Thank you very much, Humeyra.
yeah, thank you. Thank you for having me.
Thank you for listening. I hope you enjoyed this episode. To find out more about us, visit our website, thesciencetalk.com, and make sure to follow Under the Microscope podcast wherever you're listening. And if you're watching on YouTube, make sure to subscribe to the YouTube channel of The Science Talk. Thank you for listening, and we will see you in the next one. What, are you telling me that me as a human is 70% similar to the zebrafish? It's a fish. It's a fish. Are you talking about? Do you mean in our brains, like how our brains function, or do you? What does that? Okay, this is opened a completely new stream of questions now. I thought the closest we were to animals was chimpanzees. And now, this just shows how much I don't know about biology. So, for our listeners and watchers and followers, I'm sorry if I come off as this ignorant person, but really, ask me anything about nanoscience and material science, and I would be able to give you. Wait, okay, 70%. Please, okay.
That we would like to study zebrafish because it has 70% genetic resemblance to a human. that if we can study zebrafish, understand it, then I believe that it will really help biomedical people to really bring up different aspects. For example, they would mainly study the brain cells of the zebrafish, for example, to understand questions for Alzheimer's and so on. So that's why it's quite important to bring them the tools. That's our job. Bring them the tool to study it. So if we can give them the tool to study this, I believe that they can achieve really, really great things.
So cool. That is so cool.